Hear me clearly?
Yeah, yeah.
Okay. So I welcome all of you. Good evening, and I welcome all of you to this wonderful session on role of microbiome in human life, including time of embryo implantation, that is T0. And we are very fortunate to have with us our first international speaker, Dr. Sheetal Tripathi. She is the CEO of Pari Biosciences USA. And we are more than honored to have with us Dr. Sudha Prasad. And it's a real honor to introduce ma'am. And she has been such a stalwart and such a luminary in the field. So, Madam is the director of Matritva Advanced IVF and Maternity Center, which is based in Gurgaon. She has a teaching experience of more than 40 years. The ma'am was the professor and head and IVF coordinator, IVF and Reproductive Biology Center, Maulana Azad Medical College, New Delhi. Ma'am was the Dean of Faculty of Medical Sciences, Delhi University from 2015 to 16. She was the past president of Indian Fertility Society to 2020 to 22 during COVID time, and she led the society so well during COVID times. She was the past vice president of Foxy 2019, past president of AOGD, you name it, and she was there. She has a fellowship in IVF and reconstructive tubal
I
surgery.
think more than enough, Sarah. Please let me.
Let me just complete. And she started the first low-cost IVF at Azad Medical College in 2006. She received the state award by Government of NCT for meritorious service in the field of healthcare in 2003. A short-term fellowship was awarded to her by Hungary under Indo-Hungarian Educational Program. And she has more than 65 international and national publications and innumerable chapters in various books. So this was about Dr. Sudha Prasad, and we are so lucky to have you with us, ma'am. So
Thank
fortunate.
you so much. Thank
We're
you.
really honored. And we'll also introduce the moderator for the day, Dr. Monica Verma. Ma'am is from PGH Chandigarh, and she was a senior resident at AIMS New Delhi. Ma'am is heading the Sadbhavna Medical and Heart Institute, Patiala. She's the medical director of Sadbhavna IVF Plus and Sadbhavna IVF School. She has been in practice for more than 35 years now. Ma'am has held various esteemed positions such as academic committee of chairperson of academic committee Patiala Obs and Gynae Society. She was the chairperson of academic committee IMA Patiala. She was the formal secretary of Patiala Obs and Gynae Society, former joint secretary of IFS Punjab. And ma'am has been presenting at ESHRE for 6 years in a row, and this year again for the seventh year, she is going to present in London also. And she was awarded the Isar Dronacharya Award 2023 for excellence in women healthcare, awarded the Women in Business Award for Best Women Performer in Healthcare in 2023. Ma'am has a dedicated IVF skill to simulator lab, which is a one of its kind lab in the region of North India. And she's running a regular online international academic programs along with the Fertilus Academy. Her special interests are reproductive endocrinology, FET cycles, donor-induced pregnancy, and hysteroscopy. And ma'am has been the driving force behind Fertilus Academy and all the sessions which you are a part of. Thank you so much for being with us, ma'am.
Thank
And
you. Oh, thank
now
you
I
so
hand
much.
it over to Dr. Monica to introduce
Thank,
for the day,
yes.
Dr. Sheetal.
Thank you so much, Dr. Sarafi, for your kind words. And you yourself, it's all your thought process, this Fertilus Academy, the platform, and the way things are going so well. So we welcome Dr. Sudha ma'am, a very warm welcome. And ma'am has like inspired and taught so many generations of gynecologists. So we are very grateful that you are here with us. So we are very lucky, I would say, that we have an expert with us for a topic which is so much relevant in ART, and there are so many question marks regarding it. Like, we really want to know the ins and outs of microbe and microbial research. So what else could we ask for? Then we have Dr. Sheetal Tripathi. She has a track record of translating microbial research to impact. And impact, sure it is, as we can see from a very precise but very concentrated biodata. She's a gold medalist and master's from MS University Vadodara and a PhD from Dartmouth College. 20 plus years leading R&D raising to CSO. Co-created and existed 3 platforms assets. Like, I would ask you please to clarify this, because this is not our language of exited 3 platforms assets. So, but led to microbiome drugs from early discovery to phase 1, and both were acquired. She's a co-inventor and co-author on 18 patents and publications, and she has launched a DTC supplement, direct-to-consumer supplement. So she is all full of knowledge, and she's all full of stuff which we want to discuss it with you. And we are really waiting for your talk. And a very warm welcome, Dr. Sheetal. So all over to you for your very interesting overview of microbiome in human life, including time of embryo implantation, which is something which we are really concerned with, that if we can improve the ART pregnancy rates, and if we have a factor to improve it, then how to go about it. So I would request all of the attendees to put your questions in your chat box, and at the end we can have an interactive discussion. We have a few persons in our program also who are working on microbiome, so they were very keen to be with you and to discuss it. And so a very warm welcome. And as the expert, we have ma'am, who is an expert in all aspects of ART. So a very warm welcome to both of you. And we hand it over to you, Dr. Tripathi, to please start your talk.
Thank you so much.
You're the host now, ma'am, and you can start sharing your screen. Anybody has any queries, they can put in the chat box, or in the end, we'll allow you to ask questions live. Over to you, ma'am. Thank you.
Thank you so much. First, I want to thank Dr. Saraf for organizing this, and Dr. Monica and Sudha. You are such illuminaries. So in your presence, I am just very pleased to be here. Microbiome is still very much a growing space. So we still have more questions than answers. But I would try my best to share what I know so far. And as we are growing, I'm sure I would love to have more contributors in the field. So it's very nice to hear that there are others on this panel or on this call today who are in this subject. So super happy to be here. All right, I'll start my screen. Let's see. Okay, I am not on the first slide. Okay. You can all see my screen?
Yeah.
Yes.
Yeah.
Yeah, it's clear.
Okay. Great. So I thought I'll start with a sort of a broad overview, starting about what we know in general about microbiome, and then get down to how it impacts ART. So bear with me. This is an overview talk in the beginning. And when I'm in front of live audience, I try to break the ice with some initial couple of questions on what we think how the microbiome is in our body. So maybe in the virtual call, maybe we can raise our hand if you want to, or put some numbers in chat, or just think about what you think might be the answer. But what we think about the estimated mass of human microbiome in our body, we started out with some answer, and then we have revised those estimates. So if anyone wants to share something in the chat or raise your hand, but is it 2 to 3 kilogram, we think, or is it 5 to 7 kilogram, or it's 0.2 to 0.3 kilogram? And so the answer is 0.2 to 0.3 kilogram of body mass is, I see that there is one in the chat. Okay. So then going forward, okay, so the number of microbes to human cells, again, this is something that we thought a lot about in the field, that, okay, microbiome is being considered as a separate organ. So what do we think is the cell number of host cell compared to microbes? And now we think that it is almost 1-to-1 ratio. So as much as our body has about 30 trillion of cells, human cells, and microbes are about 38 trillion in our body. And in terms of estimated number of species in our body, it's about more than 1,000 species in our body of microbes. So this is pretty significant in terms of not ourselves, foreign cells that are living with us. And we already went through this, so I will not belabor this, but just to share, when Dr. Monica said that I built 2 platforms, so these were 2 microbiome pharma companies where the idea was that let's isolate gut microbes from healthy human donors and create a library of human gut microbes. And that's what we were doing at those 2 companies, where we were isolating different species. And I was lucky to be able to isolate more than 500 species from gut microbes. And again, we are still learning and growing in the field, but it was really fun to isolate some of the very hard microbes and realize that among our library, about 60 to 70% of the species were never isolated before. So it was the first time ever that we were able to isolate those microbes. And then we went on to characterize them. We were doing host microbe interaction translational based assays, and then we were figuring out how those microbes were interacting with human cells and how they could be used for various treatments. So before we go into diseases, as I mentioned that microbes comprise a huge fraction of our body. One of the most important thing to think about here is that before we even go down to the path of disease versus a healthy state, in fact, I want to bring this up, that microbes are actually essential. We have become symbionts with them, because without them, we actually do not have normal physiology. So as we look at the normal mice versus germ-free mice, we can see that for having a normal brain function, cognitive function, a robust, mature immune system, as well as the efficient metabolism, all of this is dependent on, from very early life, our microbial colonization in our body, because otherwise, if that colonization is not robust and it's not good, then we have impaired physiology in our body. So what are the niches, key niches in human body where microbes reside? The biggest organ where the large number of microbes are is, of course, the gut. But there are also other niches in our body where microbes are. And you can imagine wherever you have a port of entry, that's where these microbes reside. So there is also skin, urogenital, and mouth are the other niches. And in terms of diseases, there is now I think we can almost say that every condition for humans is somehow related with microbes, because having the right microbial community and the harmony between microbes and human body is essential for having a normal function of the body. So all of these diseases people have looked into and seen that when you have microbial dysbiosis, a lot of these indications are showing up. And this is just to show that the yellow ones are where I have worked on from therapeutic perspective. And then we talked about dietary supplement, where we were looking at heart disease and cardiometabolic disorders. So how do we learn about microbiome? And I want to just share this one example of how the field started looking into this from experimental perspective. And there is a lot in this field. So this is just one key example that I'm sharing on how the researchers went about learning about, which came first, chicken or egg situation? That do we have a disease and that is causing the microbial dysbiosis, or the microbial dysbiosis actually have active role to play in the disease and health? So in order to understand that causal relationship, this is the type of experiments that the field has conducted. So here, this is a classic paper from one of the top microbiome researcher, Jeff Gordon's lab I believe, Seattle, where they were looking at the twin study. So this is human twins. One is obese and one is lean. And what they did is they did something called FMT, so fecal microbiota transplant, where they took fecal matter from this human host and introduced that to germ-free mice. And what they saw was that from obese twin, the mice became obese. And the lean twin, the mice were staying lean. And then what they did was they co-housed the lean and obese, and they saw that the lean phenotype was transferred to the obese mice and they became lean. So this is just one example, but there are several examples in the field where it has been shown that there is this microbial dysbiosis is not just a correlation. In fact, in this case, the only difference between this folks were they were same genetics, same environment, same diet, but the difference was microbiome. And they saw that the microbiome caused obesity in recipients, and it could be reversed in diet-dependent manner. So this the field has went on to show this kind of experiments in various different human conditions. Of course, gut has been the major focus in the field because, as you can imagine, we started out thinking that that's where the biggest concentration of microbes are. So that's where we kind of the science gravitated towards learning more and more about gut in the first place. So lot of FMT work has been done, and then people have gone on to isolating microbes and creating therapeutics and using those to cure diseases in the area of gut. But there are two FDA approved therapeutics, for example, for recurrent C. diff infections in humans. And again, this is very active area of research and therapeutics development. So IBD, ulcerative colitis, Crohn's, metabolic syndrome, food allergies, all of these things are getting addressed with different ways of approaching microbial therapeutics. Similar to gut, there is also gut-brain axis that has been quite actively explored. So it's been shown that the depression and anxiety, all those neurological behaviors are also relevant with microbiota, and people have shown that using FMT again, that it can lead to significant improvement in GI as well as behavior, for example, in ASD with children. Similarly, there is, and I'll share a bit more about this, but very interesting, fascinating results are coming out from oncology field recently, where the microbiome is showing a really promising role in recovery from cancer. And one more hot topic these days is also longevity, where people have shown that FMT can, and there is this nice paper that talks about this as well, where they took FMT, fecal microbiome transplant from older versus young folks and showed that the phenotypes of aging were transferable just by microbes. So this is the oncology. There were back-to-back research papers that were recently in January, they came out, and really amazing work here. What they showed is that you have FMT with PD-1 in the non-small cell lung cancer. So one of the toughest ones to treat, they saw that the response was 80% compared to about 40 to 46% without the FMT. So again, microbiome from responders, taking that into people was helping with the cancer treatment here. And again, with melanoma, they also saw that much better response with FMT compared to the treatment alone. So how the field has thought about how do we develop, how do we use microbiome as a way to go back and treat the different human conditions. So fecal microbiota transplant is something that was started very, very long time ago in ancient cultures. And this process, while it works, it is quite challenging because, of course, there is so much variability, a lack of standardization, risk, because you don't know what that complete composition of the material is, and so on. So the other approach, again, is an ancient approach, is probiotic, prebiotic, dietary intervention. And, something to think about it, and it always fascinates me, is that every culture in the world has come up with a way to eat fermented food and different food that are actually somehow feeding our microbes and rejuvenating them. So this is the ancient wisdom that we are living with, and we are now discovering why a lot of cultures did that and how that's helpful. So this is again a really very interesting way to again take care of our microbiome. But when you think about a disease condition and trying to treat, this again lacks precision and the impact that would be needed for a therapeutic to work. So then the third approach is to create live bacterial therapeutics products that are called LBPs. And the field has thought about this from various angles. So single microbes, using just one microbe that is giving the function that is needed, or create a small community. In our case, in one of the companies where I worked at, we wanted to create something that is quite complex, almost mimicking the FMT, so to speak. So create a synthetic consortia that is highly complex, but very highly regulated, created in a regulated manner. So each strain is isolated, characterized, manufactured, and going through the CMC regulations, and then creating a pill out of that. This was a spin out from Stanford, and I was very happy to take on this project and this challenge of how do you create 150 plus microbial strains in a single pill. But the idea again here is to create that diversity and bring back that diversity that is in the gut, and reseed, so to speak, our gut microbiome. And of course, the fourth way that the field has thought about this is with the next gen therapeutics, where we engineer microbes with specific protein or pathway, metabolic pathway that is required to provide the function that is lacking in specific disease condition. So all of these are really interesting ways, and just this is a very superficial overview because the field is pretty active and very much growing. So there are hundreds of products and hundreds of companies that are working on this way of valorizing, using microbiome as a lever to bring back good health in humans. But if we look at what is the roadmap of different products that are in the FDA clinical pipeline or EU pipeline, then we can see that there are two products that, like I said, are approved, but there are also products that are in phase 3, phase 2, a lot of phase 2 and a lot of phase 1. I'm just showing some select products here. But it's really very interesting to see this field grow. There, of course, has been a lot of lessons learned as we are, learning about this really fascinating community, complex community of microbes that are living within us, in terms of not just their functions, which are not quite all we know, but learning more and more about them, how to isolate them, how to manufacture them, and of course, how to create really good, robust clinical development. Even the human study, that would be really what are the endpoints that we want to see, what are the efficacy parameters we want to see. All of that has been really very interesting in the field, and lots of learning lessons there. But we are getting there, and we hope that we will see more and more of such products coming and helping human beings as we go along. So, yeah, Dr. Sudha, do you have something to add, please?
No, I am listening, and this is definitely a new field. And in vitro fertilization, in the ART, we are getting lots of microbial thing, and endometritis is one of them. So people are doing lots of research about it, and we are in each patient we are giving probiotic to get more implantation. Of course, we exactly do not know where it acts, but yes, it
Yeah,
definitely acts and
yeah,
increases the implantation rate. Thank you.
yes. Thank you so much. Yeah, that's where I'm going next, and would love to share some about it. But we definitely think that there is a huge role that microbes would play there. And yeah, let me talk. Thank you so much for sharing that. Yeah.
Yeah.
Okay. So let's talk about microbiome in IVF. Do you see this tick mark? Okay, it went away. Okay. um, so let's first take a overview. What are we seeing in terms of fertility? The global fertility crisis, right? So it's one major reason why we are facing this issue is because we think that our prime window of prime fertility is already past when we are thinking about family building these days. And there are, of course, several other parameters. And I wouldn't belabor into this because this audience knows this way more than I do about this. But there are a lot of reasons why we are facing this challenge today. And globally, there is, the infertility rates are rising globally because of all these issues. And when we think about in US, there is 1 in 5, globally 1 in 6 who is facing infertility today, and the number of art every year is also increasing. We have hit the 1 million mark of IVF babies born each year, and again that number is rising because of that. So what we think about this in terms of where is the area that we could address, we think that today we have really good technologies to look at embryo quality. And again, I wouldn't belabor into this at all because this is the audience that is expert in that area. But we have really good tools to look at embryos today. But what we think where we are lacking or having incomplete understanding is the endometrial receptivity. So this is where we don't have really a deeply designed, scientifically designed tools that are looking at molecular level if the endometrium is ready, and if the IVF will be successful or not. So that's one of the major reason we think why today's IVF cycles are failing is because of the lack of endometrial receptivity and maternal factors, which we don't fully understand. And then there are also no approved therapies for this. So thinking about microbiome, what's really interesting is that the microbiome is modulated naturally in the way where from puberty to when the women hit the prime fertility age, we see that there is lactobacillus dominance in that age. And then as we progress in our age, that dominance is again diminishing, and then you have more diverse microbiota taking hold. So one thing to keep in mind is that the gut microbiome is opposite of vaginal microbiome. So in vaginal microbiome, we are seeing that we want less diversity as opposed to gut. For gut, for good health, we want more diversity. Here we want less diversity and lactobacillus dominance, because what's happening here is that community, that specialized community that we have been seeing in the prime reproductive age, is really contributing to the right environment, creating the environment where the embryo implantation is feasible. So what we are seeing is that the unhealthy microbiome creates an environment that is not conducive to embryo implantation. And there are very specific molecular mechanisms that are playing here, both in health and unhealthy environments, which are either supporting or destroying the receptivity. So in fact, in humans, there are several papers now and a lot of really interesting literature around this, but I'm sharing this one example here because it's a human study, and it kind of nicely shows that if you just take vaginal microbiome, look at that analysis, you can characterize the IVF patients or group IVF patients into two parts. So one is unfavorable microbiota and one is favorable microbiota. And the group went on to show that the unfavorable microbiota was able to predict with more than 94% accuracy that the IVF will fail. Whereas the favorable microbiota, that's where we get a little bit murky, in the sense that the group was able to characterize or group subgroup that group into a good or very good. And we think that at PariBio, we think that that accuracy is not enough for patients, because telling someone 50-50 is not enough for enough of information for patients to go on or doctors to go on. So, but this is very promising in the sense that we know that this microbiota is a negative factor. As a negative factor, we can really have very high prediction accuracy when it comes to success of embryo transfer. Again, our other pet peeve is also that there is no intervention beyond this today. So at PariBio, we want to address both of these issues. But that is one study, and if we look into literature, there are, of course, more and more studies coming out from all over the globe showing precisely the similar results, is that microbiome can predict failure with very high accuracy. And in fact, if you add more data to that, you can have better and better prediction in terms of art outcomes. And again, so this is something that you all can really teach me about, so I won't really get into the details of this. But what I want to share here is that, like I said earlier, the healthy microbiome, what we call eubiosis, and the unhealthy status that we call dysbiosis, they both are now known to have very clear molecular interactions with the host cells and tissue that are really impacting all stages of embryo implantation. So, yeah, and any question? Someone wanted to say anything? Okay, so I'll go on. So we know that eubiosis is indirectly impacting inflammation, but is also directly impacting the immune tolerance. and the pathogenic species are suppressing the implantation molecules and directly destroying the implantation physiology. So this is really fascinating and also something that is very tangible to go after and create solutions on this. So this is exactly what we are trying to do at PariBio. We are addressing endometrial receptivity by first assessing what is the status and then enhancing to improve the status if the patient turns out to have a dysbiotic microbiota with a therapeutic. So with that, I just want to thank all my former colleagues and thank you all for spending your late evening today and hearing this. Really, such a pleasure to be part of this community and sharing this with you all.
Thank you, Dr. Sheetal. Beautifully, fantastic talk. You have really given the very good example, the lean and the obese mice, how the FMT acts on this. So this is a burning coming out examples of microbiota. People are really thinking why the receptivity is so less about the implantation. So something else is working over here. This is very much important. And number two, the uterus, when the amnion and the chorion are there, people used to think it's absolutely sterile environment. But now we know the microbiota is already there inside, which is giving lots of balancing, and pregnancy is continuing. And once the balance has gone, it is dysbiosis. And for implantation, definitely we need the lactobacilli positive, which is the favorable one. And if it is non-lactobacilli microbiota, then it is unfavorable. This is important. This is the reason people thought about we cannot take so much of pain to do the research each patient, that yes, it is the lactobacilli, what is the number, how much is there, whether the implantation will occur in this or not. But definitely give probiotic as a empirical, you can call it, but it is definitely working. So
Yes.
this is
Yes, exactly. I think this is where we really want to understand, because we started out thinking, maybe I can share that quickly here, but we started out thinking that there are four what we call the CSTs, the community state types in vaginal microbiome. And now the field is emerging with new and new understanding about that. So there are subgroups of those, and it would be really interesting to go after this with very methodic and very clinical perspective of taking a robust study data from patients and understanding how each of these are impacting the outcome in IVF, and what are the key attributors to create something that is clinically designed product. No, we know that probiotics are helping, which is a great sign, and which is a great motivating factor that, okay, something that people are using empirically and it's working, so let's create that in a very robust manner and really make a huge impact in this field. And I didn't share, but I also went through IVF myself, had several failed cycles and then success. So, very, very deeply passionate about this field and would love to contribute to this field. Yeah.
Oh, great news. Very well spoken. I think audience must be benefited by this. Yes, Dr. Monica, you want to say something?
I just want to thank you for this very insightful and topic, which we really wanted it from the expert. Otherwise, it gets a little confusing. So we have a lot of appreciation from our doctors and appreciating your talk. There are a few questions, Dr. Sheetal, which I'll just put forth from the attendees. When you mentioned that IVF study, that it has a 94% predictability of a good outcome if the microbiota is favorable. So there, Dr. Davis, he's just asking to specify what is favorable and what does unfavorable mean when we are talking in relation to microbiota.
Yeah, Great question. We go back to that. I think so. The idea here is that they were looking at the microbial composition only. And so when you look at the microbial composition, if it is not favorable, you can predict IVF failure with very high accuracy, because you know that this is an important factor. And if it is not there, then the patients will have very low chance of success. But when it comes to success prediction, you can imagine that microbiome is not the only factor. There are many factors that are impacting success. So that is where the single parameter, looking at only microbiome alone, is not very good when you want to predict success. And that's where the team saw that the prediction was not that great. Only 55 to 65% success prediction is not that great. And this is where we want to, in fact, think about creating something, a solution that is looking not just at microbiome, but other factors that contribute to failure and success. And of course, today's day, we can use AI to really take all this data and create something that is very highly predictable, and we can implement that in a successful manner. But great question. Yeah.
Thank you so much. It sounds very interesting. We have a hand raised by Dr. Bernard. You would like to ask your query?
Yes.
Yes, thank you so much. You can switch on your video as cameras.
Video. Okay, I had to park because I was driving.
Okay.
So I'm actually doing a research on vaginal microbiome and the impact on IVF in Ghana, West Africa. And it's interesting, this topic, because there are a lot of questions, as you mentioned earlier. The first is that you are using vaginal microbiome as a predictor of the endometrial microbiome, and that's a big question to to answer when you're doing such a research, because you're looking at the vagina and it's not the same as the endometrium. It becomes really difficult. The other thing is, at what point in your FET cycles are you taking these vaginal samples? Are you taking them at the time of endometrial preparation? Are you taking them at the time of embryo transfer? Because the vaginal microbiome is so dynamic, such that if you expose it to estradiol, for example, you are expecting a lot of changes. How do you factor in all of these confounders to come out with this predictiveness of your research, whether it's a positive or negative predictive value? Thank you.
Thank you. Both were excellent questions. So totally agree that the field, there is active dialogue, or I should say discussion going on about vaginal versus endometrial microbiome. And I think there are some very interesting papers recently that have come out that talk about the cervicovaginal mucus being a strong barrier such that microbes are not freely passing through. So one notion is that the vaginal microbiome is not directly that the figure I showed is kind of a bit misleading, because the microbes are sitting right next to the embryo. But what we think is happening is that there are microbial released molecules that are going through the cervicovaginal mucus barrier, and those are the ones that are interacting with host cells. So there is really very interesting papers that have recently come out in just this year and last year, where they show that these factors or molecules that are made by microbes are directly interacting with human cells. And like I said, they are either suppressing the immune system to be overreactive, allowing the embryo to implant, because embryo is 50% non-self. So we are kind of creating that environment which is allowing that to happen. At the same time, these microbes are specialized by creating their own antimicrobial mechanisms to prevent pathogens from taking hold. So that's one way we are thinking that this vagina versus endometrium microbiome is happening. The second question about the variability in vaginal microbiome, it's absolutely true. And in fact, over the cycle, menstrual cycle, we also see that there is variability in that. So this is where what I meant by having really strong longitudinal study and understanding what are the other confounding factors like estradiol, for example, how that is impacting. So this is exactly what we would like to do, is to understand this really deeply to get an answer on when is a good time, what are the factors that are impacting when we are looking at microbiome. Yeah.
Thank you so much, Dr. Sheetal. I'll just like to ask two questions related to this, then we'll go back to the questions which we have in a chat box. So when you are trying to make this whole predictability of seeing the microbiome and AI, and you're doing so much on it, so why did you choose the vaginal microbiome? Why not the endometrial microbiome to go straight to where the embryo is going to be affected? Like why are you not going with the endometrial microbiome?
Yeah, thank you. Great question. So one is because of the biology, that the way we are thinking is working. So if it is not microbes directly and the molecules that are secreted or created by the microbes that are going through a vagina to endometrium and impacting this, then we think that it's a good way to look at which microbes are there in vagina, because they are the ones that are causing this anyway. And the second, of course, is thinking about the practical implications, right? The endometrial microbiota, we would need to do some biopsy or more invasive process, whereas vaginal microbiome is going to be a non-invasive way of doing this. So we think that there is enough scientific literature and support that the vaginal microbiome is in fact able to predict the outcomes. So this is where we are thinking is that, okay, for all these reasons, one, of course, it's grounded in biology, but also it helps with the practicality of application.
Thank you so
Can I
much.
say, I will tell you one thing. If
Yeah.
you join the IVF centers and when we are doing the embryo transfer, then the outer is, inner is pulled inside the outer, and
Yes.
outer is little cut, and then from the inner you just take the sample. So you can
Yeah.
get lots of endometrial tissue for the culture, or not tissue, rather the secretion for the culture. And probably you can do the wonderful thing, because implantation is so much of gray area that whatever we do, we are not exceeding more than 40 to 50% of the implantation of in the young, less than 35 even, so this can be done. You can
Yes.
collaborate with all IVF center and do this kind of work.
Absolutely, Sudha ma'am. This is exactly where we would want to go. You were talking about the catheter
Catheter, in
sampling.
the embryo transfer catheter, it is
Yeah.
very inner is very soft. First
Right.
you do the outer, so and then you push the inner, loaded with the embryos, and then you push it. While taking out, normally we take out like this only, but you have to ask to the IVF center that the inner should be pulled inside the outer first, and then you take out, so it is not contaminated with a vaginal wall or anything. Then you cut little outer, so it is not touched anywhere, and finally you can take the inner, secretion for the culture. So this is, you can do a beautiful work, and it will be really
Absolutely
helpful for
Yes,
all
very
IVF
helpful.
centers.
Yeah.
But I had
Yeah.
one query that have you done the obesity and the lean? Because I feel the obese patient must have the more dysbiosis than the lean patient, because the implantation rate or the stimulation to get the
Yeah.
more oocyte retrieval is also less, even though
Less.
we exactly do not know how much gonadotropin you have to give. Sometimes it flares up, and sometimes it is just few eggs are available.
Yes.
So have you checked that?
We will. I think this is one of the factors that we definitely want to look at, because as I was mentioning, the what we are not also talking is the gut microbiome here, but of course it's going to be linked. So all of these areas are very much interesting for us, and ultimately it would be this approach of understanding as much as we can. We are starting out here to be focused and understand the first step, so to speak. But absolutely very, very interesting to understand how the obese versus lean, the
Yeah,
differences. And in fact, also you can imagine that the IVF patient would also comprise of sub cohorts such as PCOS and endometritis, as you were mentioning. So we want to also understand how those microbiomes are different for those cohorts. Yeah,
good. Thank you.
very interesting.
Thank you. It's a very stimulating discussion, and we have many questions. So, Dr. Sheetal, Dr. Shrivani wants to ask how many months we need to use, I think it would be probiotics, to get proper results.
Yeah, that's excellent question. And I think what we know today, not exactly the time for sure, but what the goal is very clear. The goal is to shift from the dysbiotic microbiota to eubiotic microbiota. So we want to spend significant time so that that shift can happen. I would leave it at there. But yeah, I think this is not something that you can imagine 1 or 2 days or 3 days and it would act like an antibiotic. We don't expect to see that.
So, like when we are talking of probiotics, would it make a difference? Like we have oral probiotics and vaginal probiotics, like they'll act faster, or it makes no difference?
No, great question. And there has been really interesting studies in the literature where they have shown that there is a transfer that happens between the oral to vaginal microbiota. So eventually, I would say that would happen when you take oral probiotics. But vaginal would be probably much more efficient route of delivery for this.
Thank you. Thank you so much. So we have Dr. Lucky asking, he has two queries, Dr. Sheetal. One is that what advice will be given for a probiotic? Will you give probiotics for gut patients who have gut inflammation? And secondly, is it possible that the gut can alter, the microbiome can alter the embryo transfer results?
Yes, I think as we discussed, both correct, because like I said, there is notion that the gut microbiota also transfers. So lactobacillus seeding could happen from gut microbiota, and that is the oral probiotic. So yeah, to answer, both are true, that yes, the treating gut dysbiosis would help in that sense and also help the seeding.
Thank you. Thank you so much. So we have another question from Dr. Bernard. He is asking that, are you looking at the impact of various lactobacillus species and its impact on IVF success, as we have some species which are associated with poor outcomes, like especially lactobacillus iners? So
Yeah.
when you are doing your microbiome studies, are you looking at the species, subspecies specifically?
Exactly. Yeah, we would go down to strain levels, because,
Yes.
even within the same species, all the strains will not have the same genomic makeup and the phenotypic characteristics. So we would have to get down to really understanding how that, and not just the genomes and the phenotype of the microbe itself, but also looking at host microbe interactions and seeing how that are modulated with certain strains versus not.
Thank you so much. Dr. Sheetal, I'll just ask you your opinion about I go back to the endometrial microbiota. We have a few studies which say it's a lactobacillus predominant. Then we have a few studies also who have done it by taking it to a laparoscopy that it is not lactobacillus dominant. And considering the pH of the uterus, which we know is like the embryo culture is 7.3. So what are your viewpoints on the gross microbiota of the endometrium? Would you consider it lactobacillus dominant or not?
I would say, yes, this jury is still out on this one. What I would say is that the challenges are coming from twofold. One, like we were discussing with Dr. Sudha, what she was describing is very, very precise way of taking the sample. And if that's not done that way correctly, then you could imagine that there are ways of contaminating that. And the second fold of challenges coming from the fact that already the amount expected for the microbial load in that area is very low. So we expect only 10 to 2 or 3 when it's coming to endometrial environment. In vagina, there is a lot more biomass compared to endometrial biomass of microbes. So I think for those two factors contributing to why we are seeing so much variability in the literature when it comes down to this. So again, I'm waiting for this large study that would really show us conclusively that what is really happening here. What I would say though is based on this CVM and how the porosity of CVM is discussed in the literature, it seems that it should mimic what is happening in vaginal microbiome space. So depending on if the patient is in healthy status and has lactobacillus dominance, then you would see more of that.
Yeah, I think fantastic.
Thank you. Dr. Sheetal, it was a really we were really looking forward to it, and it's a really nice discussion which we've had. So anybody would like to give your comments or raise your hands or questions before we have our final comments from the experts?
So it was a
we just
fantastic
have appreciation left for you.
It is very well appreciated by all audience.
Yes.
And it's a really, you have really taught in a very nicer way, I must tell you, that the way normal audience also can understand what exactly the microbiota, because we talk about the dysbiosis and microbiome, we exactly do not know, unless otherwise we will go through this whole stage. So very beautifully, excellent.
Thank
and
you.
Need
Thank you.
really more research about the microbiota, that from the childhood to the menopausal, the microbiota is important. There should be balanced, and as soon as there will be imbalance, the disease process starting from the PID, from the cancer cervix, or anything, it can happen. So it is very much important, not only in the reproductive age group, in any age microbiota balancing is important. So nice.
Yes. I can't speak enough of that. I think it's so, like I said, from very early childhood all the way to 100 years. If you look at the microbiome of centenarians who are 100-year-old, it's so different from other folks, and it's really fascinating. And something I didn't talk about, but also very interesting, is that from early on, when the babies are breastfed, there is fascinating new studies that are coming up, which shows that there is both ways interaction. So human breast milk is designed to create the right microbial niches for baby's gut to be seeded, but there is also baby's gut giving feedback that, hey, this is what I have. I mean, not in this words or communication, but
Yes.
molecular communication, that this is what I need and this is what I have. So it's really interesting how we have evolved, co-evolved, we should say, with microbes.
Thank you. Thank you so much.
Thank you so much.
So
Dr. Bernard has another query. Just wants to know that the research which you're doing, is it specifically for frozen embryo transfers or fresh or both?
Yeah, great question. We are starting with both at this point, but as like I said, as we grow and we collect more and more data, we will start to see the delineation between those two.
Yes.
So very good question.
So we are really looking forward to the answers you're going to give us, Dr. Sheetal. And it's been so nice to connect to you.
Dr. Sheetal, I have a question. Anyway, we can, modulate vaginal microbiota. Like you showed the first experiment of, transplant of
Yeah,
fecal microbiome. So anyway, we can pick up some vaginal microbiome from a favorable vagina, which has shown implantation. I mean, it's just a vague thought I had. I mean,
yeah.
can we
No,
put
no,
it?
no.
It's an actual
Yeah,
thing.
great question. And there is something called VMT. I didn't talk about it, but it's vaginal microbiota transplant, and people have done that and showed that there is really good impact. So there have been case studies where there were recurrent pregnancy failures, and people have taken healthy vaginal microbiota and transplanted and seen success with that. So this field, like I said, is growing, and if we had as much focus as gut microbiome from the beginning, I would say in past 10 years, we would have known a lot more. But we are coming to it now anyways, so which is a very good thing. And we are starting here, and more people, if they take interest, I think we would learn a lot more, a lot faster. But great question. Yeah.
Thank you.
Thank you so much.
Thank you. I
Thank
think
you.
thank you so much, Dr. Sudha. It was an honor to have you, and it is always nice to hear your input.
I also enjoyed with the whole discussion. It's fantastic.
Thank
Thank
you so
you,
much. Appreciate your kind words, ma'am.
Thank you. And thanks to Dr. Sarappreet for having us all
Yes.
here together and making
It's an
things
honor.
happen.
It's an
And
honor.
making things happen. And it's so wonderfully done. The academics, the organization, the planning, everything. He's a great person and a great clinician and an excellent, I always call him, he's a magical embryologist. So very good. So,
Yes.
and Sheetal, it was really nice. It was a topic like which we always wanted to hear it from the expert, because reading it from books, it gets confusing. Reading papers, it gets confusing.
I
So
know.
we look forward to being with you again after some time, and we'll have something new from you and to enlighten us how it's going in IVF. Thank you so much. So
Thank you so much. Really, really appreciate it.
Thank you so
Thank
much.
Have
you.
a great evening, everyone.
thank you. Thank you.
Thank you.
Thanks to all. Thank you. Good day.
Good night. Good night. Thank you.
Bye bye.